Hi, good morning, everyone. Thanks once again for accommodating me and switching to online learning for this week. So this session is being recorded. If you need to revisit some of the concepts that was discussed today, you can feel free to watch that. And for those of you who missed it, you can just watch the recording as well, although I assume those of you would not be here right now. So let's start by going through some of the things that we learned last Monday before we dive right into today's lesson. So if you have your study guide, please turn to this diagram. It should be on the last page of the study guide. I've also turned on live caption. Okay, so for those of you who find that helpful, you will have that option on Zoom. If you have any question during the session, just either unmute and ask or type your question in the chat box. So here is the diagram that summarizes everything that's happening in the process of DNA replication. And I just want to remind you that the things that we learned, the steps that we learned regarding DNA replication, that is for the eukaryotic cells, okay, for prokaryotic cells, rather, for simple cells like E. coli. And we're going to see how that differs from what's happening in us in eukaryotic cells. But first, let's summarize everything that we've learned up to this point. So here we have two strands of DNA, and we are looking at a replication fork, which is one side of the replication bubble, right? So you might remember we have this bubble right here, and one of it is going to be like 5' to 3', and then the other one is, of course, anti-parallel, and we are focusing on one side of the replication fork. All right, so in order to create this origin of replication, we're going to need an enzyme to do that. Okay, and that will be enzyme number 2 right here. Does anybody remember what is the name of that enzyme? Number 2 right here. That's right, Andrew. That is the helicase. Okay, so the purpose of the helicase is to unwound the DNA, unzip it, right? And it's going to continue to unzip it towards the fork, and so you can gain access to the parent DNA. But as helicase is unzipping the DNA, it creates tension within the double strand, and we must relieve those tension to prevent it from breaking. And that's why we're going to need another enzyme here, which is the gyrase. Okay, the numbers are not really referring to the enzyme themselves. The numbers are supposedly corresponds to the steps of what's happening. So don't worry too much about them. But basically, you have to relieve the tension as the helicase is unzipping the DNA. Now, the two strands of DNA, they have a tendency to come back together because they're attracted to each other, right? And we do not want them to come back together. We want to prevent reannealing. That's what we said in class, right? And that's what these little circles are. They will bind to the separated strand and prevent them from coming back together. We call these single strand binding proteins. Okay. And in order for replication to start occurring, what we need to do is we actually need to lay down a few nucleotides, right? So we're going to put a few RNA nucleotides here. We call them primers. Okay, so we're going to put a few primers here, and then we're going to put a primer here. Now, on one of the strand, you only need one primer. Okay, so this thing right here is called the RNA primer. It's going to be complementary to the parent strand, but of course, being RNA, you're not going to have the thymine. Instead, you're going to have the uracil instead. Okay, so you're going to have another primer right here, RNA primer, and all these are primers. Okay, so the enzyme that lays down the primer is shown here by this square. Anyone remember the name of the enzyme that creates the RNA primer? That's right, Pavisha. That is the primase, the RNA primase. Okay, and again, the reason we need to have the primer is because DNA polymerase 3 is not able to initiate the process of replication. Okay, it doesn't know where to start, right? So the RNA primer kind of, help it position itself and start the process. All right, and that's what this thing is right here. I mean, they're all oval shape. I'm not going to ask you to label these things right on the test. There's no point, but it's just here to summarize everything that's going on. So this thing right here, that is the DNA polymerase 3. Okay, and it can only create a new strand in the direction of 5' to 3'. So you see 5' here, and it's just going to continue to build it all the way towards the fork. Okay, whereas over here, you build a little bit, you build a little bit, and then you build a little bit like that. Okay, remember, as you unzip the fork, you will need to have another primer here and another primer. Right? So this bottom strand that is not being synthesized continuously, we call this the lagging strand, right? Lagging strand. Okay. So lagging strand, a couple things you should know. It's again discontinuously. There is these Okazaki fragments, right? It's away from the fork. Okay. But it's still nevertheless 5' to 3', right? Okay. 5' to 3'. If you look at the new strand that's being built, right? That is 5' to 3'. 5' to 3'. Okay, the directionality does not change. Okay, what changes is just whether it's being made continuously or whether it's being made discontinuously. So that's lagging strand, and up here, the one that's being created continuously, that would be the leading strand. Leading strand. Okay, so continuous, and also 5' to 3', and towards the fork. Okay. So it's going to keep on going. Here we have another DNA polymerase 3. What's not shown in this picture is what happens as the DNA polymerase 3 approaches the primer. So I mentioned this in class, right? Number 3 will fall off when it gets to the primer, and then the DNA polymerase 1 will take over. Okay? So DNA polymerase 1 will remove all these RNA primer and replace it with the appropriate DNA, right? And finally, we're going to glue them together with another enzyme, okay? And that is shown here by this thing. That is the DNA ligase. Okay, it joins all the fragments together. Okay, so that's basically everything that we learned last Monday. Any questions before I go back to the PowerPoint slides? All right, fantastic. So that's the summary right here. It's the same diagram. You can match it up yourself. There is one additional point here that I did not mention last week, and that's point number 3 here. Okay, both DNA polymerase 1 and 3 has this proofreading capability. Right? When they're making the new strand, it's like typing on the keyboard. Sometimes you type and then you realize you made a mistake and you just automatically press backspace and correct it, sometimes without even thinking about it, right? Because it's all muscle memory in some ways. And for DNA polymerase 1 and 3 is exactly like that. If they detect an error, if they put a T instead of a G, for example, and then the nucleotides don't match up, they will remove the error and then replace it with a new one. Okay, so this proofreading ability is very important because we want the DNA that is being copied to be exactly same as the original, right? But you know, it's not perfect, so sometimes errors do occur, and the mistake is not corrected, and that will result in what we call permanent changes in the DNA sequence, and that's going to be mutations. Now we're going to talk more about mutations next lecture number 7. We have a whole section on it. But in our body, we have two types of cells. We have somatic cells, okay, and then we have germ cells. Okay? Somatic cells are basically all cells in the body except for sex cells. Okay, except for sperm and egg. Whereas germ cells, they are the sex cells. They are the sperm or the egg. So if you have a mutation in the somatic cell, it will only affect you. Okay, you will not pass it on to the next generation. But if a mutation occurs in the germ cell, in the sperm or egg, and it turns out that egg and sperm gets fertilized and create the embryo, then it will affect future generations, right? Okay, so mutations in somatic cells will only affect you as an individual, but germ cells possibly can pass on to the next generation. Right? So you might hear people say gene therapy, which is something we'll learn in lecture 9. So there's a debate, right, of ethical debate about modifying germ cells as opposed to modifying somatic cells. If you do gene therapy on somatic cells, it will only affect you as an individual. But if you do it on germ cells, right, then potentially you affect many future generations, right? So keep that in mind because we're going to come back to this concept for the remainder of the course. You'll hear somatic cells and germ cells a lot. All right. Here is a picture of a diagram that shows how ligase work, right? We have the parent strand here that's shown in darker purple. And here we have the DNA's polymerase 1. They didn't say it, but it's 1. And I know it's 1 because the primer here is being replaced, right? It was red before, it was RNA, but now it's replaced with actual DNA. But you still have these gaps, right? That needs to connect the nucleotide together to form the sugar phosphate backbone. And that will be the function of DNA ligase. Okay, seals the gap. So that's DNA replication in prokaryotic cells, right? In bacteria, E. coli, something like that, right? It's very rapid. And it happens quite fast. E. coli, they can divide, you know, in 20 minutes or so, they're able to complete the whole division. That's because they do not actually have that many bases, right? And we talk about how the entire E. coli genome, it's about, you know, 3 to 4 million base pairs, and whereas humans, we have billions of base pairs, right? So it takes a much longer time for our cells to divide. So the DNA replication process, the ones that we've learned so far in prokaryotes, is very similar to what's happening in eukaryotes, in us. Okay, but there are a few key differences here. So let's take a look at 3 main differences. Number 1, eukaryotes have linear chromosomes. Okay, as opposed to circular for prokaryotes. You might remember I draw a picture of the bacterium way back in lecture 3, right? They have these circular DNAs, and then they also have these plasmid. But all these things are a circle. It just goes around in a loop. Whereas us, we have linear chromosomes. Okay, a total of 46 of them, and they're very long. So what that means is it will take a very long time if we only have 1 or 2 origins of replication. Okay, so to overcome that problem, we will create, you know, up to 100,000 of these origin of replication. So in prokaryotes, there are very few origin of replication, but for eukaryotes, we have many more. So here is a picture that I drew on the board last time. I don't know if you remember. There's a parent DNA, and then we have multiple bubbles, multiple origins of replication. And as the bubble expands horizontally, right, they will fuse together with other bubbles, and ultimately you are going to have 2 new strands. Okay, so that's the first difference. Number of origin of replication. The second difference is that most of the enzymes in prokaryotes have a counterpart in our body, okay? But some are represented by multiple enzymes. So instead of having DNA polymerase 3 and 1, right, in humans, we actually have 5 different DNA polymerase, okay? So they have overlapping functions, but you know, at the end of the day, right, the DNA polymerases, they are there to create a new strand using the parent strand as a template. Basically, it's the same process, but there are just more variety to the enzymes. Okay, and this last point is an important point. Okay? Some DNA are lost during each DNA replication in eukaryotes. This doesn't happen in E. coli. This doesn't happen in bacterium, in prokaryotic cells. It's only eukaryotic cells that have linear chromosomes that would experience a loss of some DNA every time they divide. So I'm going to explain to you why that happens. Sorry, just give me a second. All right, so maybe you want to draw along to try to understand why this happens. So here we have, again, like 5' to 3'. It's a little bit too thick. Give me a second. 5' to 3'. This is the one of the parent strand. And of course, on the other side, we're going to have the 3' to 5'. Okay. Now, same story as before. Okay. So we're going to have 1' here. And then the new strand is going to synthesize all the way to the end like that. That's easy to understand, right? Because we are going from 5' to 3' on the new strand. So this is the leading strand, as you know already. On the other side, we have the lagging strand, right? And we would have to put a primer. And then as you open up more, you're going to need another primer and another primer and another primer, right? It's the same story so far, but stay with me. You're going to see what the problem is. Okay? DNA polymerase 3 will replicate this way, replicate this way, replicate this way, replicate this way. Again, so far, everything is exactly the same as what we've discussed. Now, what happens to this little bit of RNA? Well, when the DNA polymerase 3 gets here, it falls off, and polymerase 1 will come and replace this with actual DNA, right? We saw that happening. Same thing here. This little chunk will get replaced by the polymerase coming downwards, and this will get replaced. However, we have a problem with this little bit. Okay, there are no upstream polymerase, right? There's no DNA polymerase coming from further up because we're at the end of the DNA, we're at the end of the line, right? So with no polymerase coming down from upstream, there's no trigger for polymerase 1 to bind onto. And so this little bit of RNA is never going to be replaced with actual DNA, and it will just get lost in the transcript in the DNA replication process. Okay, so this segment of DNA will be lost. Does that make any sense? Do you guys understand? That's not going to be a problem for here because we're going all the way to the end here, right? Okay, but eventually it will be a problem though. You might remember I said in class that the leading strand on the other side becomes the lagging strand, right? So, you know, as you open up more, you're going to need another primer and another primer and another primer. And so on the other end, you know, this little chunk here will not get replaced either, right? So what that means is you start with DNA maybe this long. After one round of replication, you're going to chop off a little bit, right? So it's going to get shorter. right? Because these will not be copied for the reason I have explained. Does anybody have any question about this? It's a little tricky to understand that sometimes. Okay, so this is not a problem for prokaryotes, right? That's because prokaryotes have circular DNA, right? So their DNA goes in a loop. For simplicity, I'm just going to draw one strand. So let's say you put a primer here, right? Okay. It's going to keep on going, and then it's going to loop back on itself, and you will always end off at where you were started, right? And you will always be able to replace the primer because you make one full circle. But for linear DNA, the ones that we have, we don't have the luxury of doing that. So we will end up losing a little bit every time we divide. So our DNA started with this length, and it's just going to get shorter and shorter and shorter and shorter every time we divide. We okay with that? All right. So what do we do? I mean, like, is that okay to lose a little bit of DNA? Is that going to affect us? Well, it's not too good to lose DNA, as you can imagine, right? Let's say this is your DNA, and then we have a gene here and a gene here. Some genes is what encodes for the protein, right? So you might have like gene 1 here, you might have gene 2 here, gene 3, right? They're not necessarily like connected directly, right? So you might have a gap, and then you might have like gene 4 here or whatever. Now, if you lose a little bit of DNA every time, you will start to lose genes. You will lose a little bit of gene 1. Eventually, the whole gene 1 is erased, and the whole gene 4 is erased, right? So we don't want that to happen. That's why we have something called telomeres. Okay, telomeres. They are found at the ends of linear DNA. Okay, and they are just repeats of TTA GGG. So at the end of your chromosomes, you will have a bunch of TTA GGG. Okay, TTA GGG. It repeats like hundreds and hundreds of times. TTA GGG. Okay, so what happens is, when you do lose a little bit of DNA at the end of each replication, you will just erase a little bit of the telomeres, okay? And your genes remains intact. Next time you erase a little bit again, right? So it postpones the erosion of genes that are close to the ends of the DNA. Okay, and as long as you have telomeres at the end, your DNA will be okay to be copied a few more times. But eventually, you will eat up all the telomeres, right? And at that point, you are going to have critically short telomeres, and the DNA will not be allowed to replicate anymore. Okay, so here in the picture, we can see these are all the chromosomes, and the part that lights up at the end, that's where the telomeres are. And you would expect them to be at the end, right? Because the problem occurs at the end. They're kind of like that plastic thing that's protecting your shoelaces. And if the plastic things fall off, then your shoelace starts to fray and break and unwound, right? And that's what telomeres does, okay? If they are too short, if they're not there anymore, then the DNA would just break apart, and that will be not good. So when the telomeres becomes critically short, the cell will not be allowed to copy their DNA, which means they're not going to be allowed to divide, right? Remember, DNA replication precedes the actual division, right? If you can't copy your DNA, you're not allowed to divide. Okay, so most cells can divide, like 40 times ish, and then they will not be able to divide anymore. They enter what we call a senescent state. Okay, senescence. That's when the cell have divided too many times. Their telomeres are too short now. They will not be allowed to divide further. Okay, so there's a theory that goes like this, right? If there is a large enough pool of cells that are senescent, then it contributes to the process of aging. Okay, and we lose the ability to heal fast. We our metabolism slows down, and so on and so forth. At the heart of it, people believe it's because of an increase in the percentage of cells in the body that has entered senescence. All right. So do we all understand the relationship between DNA erosion and the telomeres to protect the ends, as well as what happens when the telomeres are gone? Any questions on that? It's harder for me to gauge whether you guys are with me. In class, I can see your faces, and usually I have a pretty good read of what's going on. But here it's much harder. So if you do have questions, please do not hesitate to ask. Thanks for the feedback there. Okay, do you guys know Dolly? Anybody have heard of Dolly before? Maybe, yeah. Orion, you heard? Yeah, Dolly. Okay, so Dolly is if you didn't know, it's the first mammal to be cloned. Okay, like identical copies of the donor, right? And it uses what's called a nuclear transfer, a process called nuclear transfer. So let me just quickly explain to you what happened there. So actually, to create Dolly, 3 separate sheeps were needed. Okay, so we have an egg donor. So they take an egg from a donor sheep. They actually remove the nucleus. Okay, so remove nucleus, and then they have a nuclear donor. Nuclear donor. Okay, so they take a somatic cell from the donor. Somatic cell, and then they remove the nucleus. Remove nucleus. Okay, and then they put the nucleus inside the empty egg, the egg that doesn't have the nucleus anymore. So then what you get is an egg with a donor nucleus, right? I should maybe shade this in to distinguish it. And then now they put it in a surrogate mother, right? To put it in the embryo, in the uterus of the surrogate mother, and then it gives birth to the dolly, basically. Okay, so 3 sheep is involved: the donor for an egg, a donor for the nucleus, and then the surrogate to actually grow the sheep. Okay, so what happens is it made a big news because it was a scientific achievement, but the sheep only was able to live 6 years, okay, half of the expected lifespan. Why the short lifespan? People who's not familiar with the scientific concepts might wonder, right? Is it because the cloning process actually creates a sheep that's unhealthy, or is there something else? It actually has to do with the telomeres. Okay, so as I was telling you, right, the whole class basically, every time we divide, our telomeres get shorter and shorter and shorter. Okay, so when you were born, you have telomeres that are really, really long. But now in your 20s, in your 30s, in your 40s, your telomeres are much shorter now. So when they take the nucleus from the donor sheep, that sheep is not in its infancy, right? That's like an adult sheep. And that sheep, let's just, for the sake of discussion, let's just say that sheep is 10 years old. So the telomeres in that donor has eroded for 10 years already. So when they transfer the nucleus to the egg and ultimately became the clone sheep, the sheep is born with a cellular age that's equivalent to 10 years old, right? Because the telomeres that it inherited was 10 years old from the donor sheep. Do you understand? Right. So if they were to do it for like a human, right? If they take my nucleus and then they transplant and then they grow like a mini Vincent, the baby Vincent that came out would have a cellular age of 40. Okay, because that's like the age of my cells, and has been eroded for 40 years already, right? So they will have these critically very short telomeres, and once they divide a few more years, they will become critically short, and eventually even though it was only born for like 2 or 3 years, but then cellularly it would be very, very old, and that's why they didn't the sheep was not able to live very long. Do you understand the relationship here? So here, right? This is what happens in nature. Okay, if you're not like tampering with cloning and stuff like that, when we make our sex cells, the sperm and the egg, when we make our germ cells, there is a special enzyme called telomerase. Okay, and the function of telomerase is to re-lengthen telomeres. Okay. So even though you're like in your 30s, in your 20s, your telomeres are short, but all the sex cells that you produce, all the sperm and egg, because of this enzyme, the telomerase, they will reset the telomeres. Okay, so that when you give birth to the next generation, they don't start with the same length at the time you gave birth to them. Okay, when you give birth in your 30s, your baby is going to be born with brand new telomeres. Okay, thanks to this enzyme, and this enzyme is supposed to only activate in germ cells. Okay, this wasn't happening with the cloning because they took the nucleus from the somatic cells, right? There is no telomerase in the somatic cells. There is no resetting in those lengths. So they inherit, the sheep inherited the shortened telomeres. Okay, but that doesn't happen in real life because we have the telomerase in the germ cells. Okay, so with telomerase, you maintain the telomere length. Without it, it just gets shorter and shorter and shorter. Eventually, it will be critically short and you enter senescence. Okay. Cancer, which we will come back to in later slides, it's essentially uncontrolled cell division. Yeah, you have a cell that has gone rogue and just keep on dividing and dividing and dividing. And I told you about HeLa cells, right? You might remember that, right? It's the cancer cell that was taken from this patient with ovarian cancer, and they were able to grow it in a petri dish indefinitely, right? So we call them immortal cells, right? And you might wonder, well, how is that possible? I thought if you keep on dividing, then your chromosomes will get critically short, and there should be some kind of mechanism to stop it from dividing further, right? So that's what happens in normal cases, but in cancer, they have found a way to activate the telomerase. Remember, telomerase was supposed to only be found in germ cells, right? But these cancer cells, they were able to turn on the production of telomerase and gives them the ability to re-lengthen the telomeres. And as such, they can keep on dividing without the erosion. That's why they are immortal in that sense. And a lot of researchers are trying to explore the idea of inhibiting telomerase as a potential treatment for cancer, because the idea is if we can shut down the telomerase in these cancerous cells, then the telomeres will start to erode and they will become critically short. And when it's short enough, it should stop the cancer from dividing altogether. Right? But you can kind of understand, you know, the side effects of that, right? Someone who uses telomerase inhibition might end up being not able to have kids, right? Because then the telomerase would be affected in germ cells as well. But it's still in its very early stage of research. There's no actual telomerase inhibition therapy out there yet. So there you go. The relationship between DNA replication and telomeres and telomerase. All right, let's do some pollings. Much easier to do polling question here on Zoom than it is to do it in class. So you should see a polling panel that came out. Let's try to do this. All right, let's take a look at the results. So first one, prokaryotes have telomeres and eukaryotes do not. That is actually the other way around, right? Prokaryotes, they do not need telomeres because they have circular DNA, right? It is us who has linear DNA that needs the telomere. So that's not the right answer. Prokaryotes chromosomes have single origin of replication, whereas eukaryotic cells have many. That sounds about right. But let's check the other ones. Prokaryotes produce Okazaki fragments during DNA replication, but eukaryotes do not. We both do. Okay, both of us will have the Okazaki fragments. And primers are needed for both prokaryotes and eukaryotes because the same situation occurs. The polymerase 3 does not know where to start. Right? The primer will help us position them. So the correct answer is B. Let's try another one. This one's much easier, hopefully. Okay, thank you for participating. That is the ligase. That's correct. Okay, the ligase will connect all the broken pieces together at the end. I think we have another one right here. Okay, very good. The primers are removed by polymerase 1. Last one. Okay, I think this one is causing some divide in the class. Okay, so telomerase, first of all, is an enzyme, right? This is an enzyme. And it's only supposedly normally found in germ cells. So in cells that becomes the sperm and the egg. And the purpose of the telomerase is to re-lengthen the shortened chromosomes in some cells, okay? Is to re-lengthen the shortened, we can replace this with telomeres, right? Okay, re-lengthen the telomeres in the germ cells. Right, that's what the sum cells is talking about. So that's the correct answer. Does it prevent cancer development? No. Okay, telomerase actually is what enables the cancer cells to keep on dividing, right? Targeting the telomerase inhibition, that's what prevents cancer development. Okay. Does it remove non-coding repeats? No, it actually adds, right? To re-lengthen the telomeres, you need to add the TTA GGG. Right? It doesn't shorten the ends of chromosome, it lengthens it. So these are all wrong. That's why it's not all of the above. Okay, so telomerase is the enzyme that re-lengthens the telomeres in germ cells so that the next generation will not begin with shortened telomeres. Okay, we don't want to end up like the dolly, right? Okay, born and then age very rapidly cellularly. Okay, we want to reset the telomeres when we have kids. All right, hope that makes sense. All right, moving on. Next, we have the cell cycle and mitosis. So you might remember cell theory from way back in lecture 3. Right? Let's just quickly revisit it. Every living organism consists of one or more cells. And cell is the smallest unit of life. Individually, they are alive, right, as much as the whole organism. And then there is one part that says all living cells come from the division of pre-existing cells, right? Okay, so that's what we're focusing on for this part of the lecture. This is basically talking about the mitosis, the actual cell division. And the first half of the lecture, we were actually talking about this last point. Cells contain hereditary material, which they pass to their offspring during division, right? And in passing on their genetic material, they must first clone all of it, right? They must first copy all of it. And that's what the DNA replication was about. So now that we have copied everything, how does the actual division occur? So why do we divide? What's the purpose of cellular division? In general, we have a couple of reasons. Number one, to replace lost or damaged cells. If you have a cut, you've damaged some cells, and you might even have lost a few, and those must be replaced. And where do we get new cells? Well, we have to do cell division to make more new ones. Growth and development, one of the characteristics of living things, right? The number of cells you have in your body now is certainly way more than what you had when you were just a baby. All those new cells came from the division of pre-existing cells. And for some organisms, cell division is actually a way of reproduction. Okay, human reproduction is sexual, which means you need two separate individuals. One contributes half the chromosome, the other contributes the other half, and you combine to make a new individuals. But for many organisms, the type of reproduction they have is asexual reproduction. Okay, and they use mitosis or cell division as a main way of creating more of themselves. So things like starfish, amoeba, many plants, and there's some single cell organisms like this thing called a hydra. They all reproduce asexually. Okay, they only need one individual, so that's an advantage, right? If you're the only one, if you only sea star left in the ocean, you will still be able to make more of yourself by this process of asexual reproduction. You do not need a partner, right? So it's much easier to reproduce compared to sexual reproduction, very fast and require less energy. Okay, so in this process, once again, right, there's no fertilization of egg by a sperm, right? You only need one individual. The disadvantage is low genetic diversity because you're just cloning yourself, right? Every single sea star that came from the original one will be identical copies of itself. So with very low genetic diversity, what happens is if there is some kind of environmental changes that target a particular set of genetic makeup, then you can wipe out the entire population very quickly. Let's just say, for the sake of argument, right? Let's say this sea star is very sensitive to low pH. Okay, very sensitive to low pH. Okay, and it makes a lot of clone of itself. Okay, like you have a whole population, a whole colony made up of like 1,000 of these things. Everything is genetically identical because they came from sexual reproduction. And then, for example, because of sudden climate change, the ocean's pH drops substantially. Then all the sea stars are going to get wiped out because they are all susceptible to low pH, right? There's no genetic variation within the population. There's no room for evolution that we talked about, right? So that could be a big disadvantage. But it's not all, gloom and doom. We also learn about the fact that rapid division can be a way to introduce genetic diversity. It might sound like, a contradictory, right? How do you create genetic diversity if all you're doing is copying the same thing? But we talk about how mistakes can occur, right, when you do DNA replication. Right, the DNA polymerase 3 and 1 are supposed to check for these mistakes, but when you do it so fast for so many generations, it's inevitable that sometimes you will create mistakes. You will have mutations, and if some of these mutations are advantageous for a particular environment, then it could lead to evolution of the population. So, this disadvantage is sometimes under the right condition also an advantage as well. Okay, but we don't do this, right? Humans, we don't do asexual reproduction. Like I said, we do sexual reproduction, and we rely on a separate process that we will learn later on in lecture 8. So just to recap, this whole concept of DNA and chromatin and chromosome, because it's very confusing for many people. So DNA is the macromolecule that makes up the chromosome. So in its very native form, that's not wound up, just the molecule itself is the double helix that we are familiar with, right? You see the sugar phosphate backbone here, and these little bars, they represent the ACTGs. But these are very, very long molecules, and there's no way we're going to be able to pack all that inside the nucleus. And that's why we have to wrap them up in these proteins called the histone proteins. The name is not important. Just know them as like packaging protein. And when you wrap them all up, they become more condensed. And this condensed version of the DNA, along with the proteins, we call it chromatin. Okay, chromatin, right? So protein, that's not how you spell protein. Protein plus DNA gives you the chromatin. Okay, you see the IN here, chromatin, protein. That's how you remember it. All right. And then just before the actual cell division, we want the chromatin to condense even further, right? So it's easier to drag them around in the cell and move them around. So when you condense it even further, you get chromosome. So essentially, they're all the same thing. They're all just DNA, but the level of how much they're compact, how much they're condensed, the level of coilness, that's what distinguishes them. In the most coiled form, that's the chromosome. In the most uncoiled form, that's the DNA. So normally in a cell that's not dividing in the nucleus, you will have the chromatin. Okay. And then when it's dividing, you will have the chromosome, which is the most condensed form. Chromosomes in humans, different organisms has different number of chromosomes. You probably knew that. And humans, I've asked you guys multiple times in class already, and most of you told me that there are 46 chromosomes in total. And they comes in pairs, right? The 23 pairs. So 23 from mom, and 23 from dad. Okay. And they are numbered based on size, right? So everybody would have 2 pairs of chromosome 1. That would be the longest one. So chromosome 1 from mom, chromosome 1 from dad. And then you have chromosome 2, which is a little bit shorter. Chromosome 2 and 2, 1 from mom, 1 from dad, so on and so forth, all the way up to chromosome 22. Okay, 22 from mom, chromosome 22 from mom, chromosome 22 from dad. And then these chromosomes, they do not play a role in determining the sex of the individual. So we call them autosomes or autosomal chromosomes. Okay, so they determine everything else about you. Your hair color, your blood type, whether your earlobes are attached or whether they are dangling, right? All these other things about you is encoded within the autosomal chromosomes, okay? The last pair, number 23, that's the sex chromosome. And in female, it is XX, and in male, it is XY, okay? So who is responsible for determining the gender of the child? Is it the father or is it the mother? When I say responsible, I don't mean they get to choose, okay? Like nobody can choose what the gender of the kids is. But genetically, who's responsible? That's right, the father, right? The father, XY, right? Because if the father pass on the Y, and you're going to have a son. If the father pass on the X, you're going to have a daughter, right? It's the male that's responsible genetically for the sex of the kid. I actually have been trying to explain it to my grandmother for a long time, because sometimes she's very traditional, right? So she would ask me, how come your wife only gives you daughters, right? Where's the son? You have to have more kids and have a son, and I had to tell her it's not my wife's fault, Grandma. It's actually my fault. I'm the one who is supposed to pass on the genetics, but she actually doesn't really quite quite get it. But yes, it is the man that's responsible for determining the sex. So that's the sex chromosome. Okay, the other 22 pairs, that's the autosomal chromosomes. And numbers of chromosomes you have doesn't actually corresponds to the number to the size of the organism. A lot of organisms are bigger, but they have like less chromosomes, and some are like smaller. Like goldfish is a lot smaller, but they have a lot more chromosomes. So there tends to be no correlation between the size of the organism and the number of chromosomes they have. All right, so now we can talk about the cell cycle. If you think about like a cell, doing its own thing and then ultimately dividing to become two, the cell actually spends most of its time in what we call interphase. Okay, so in interphase, the cell is just performing its normal function. Right? If it's supposed to be detoxifying, then that's what it's going to be doing. Okay, so if it's supposed to be making hormones, then that's going to be doing it in the interphase. Okay, so in the interphase, just nothing special, just doing its job, and growing in size, getting bigger, and gathering nutrients and resources in preparation for the eventual division. Now, of course, not all cells are going to be able to divide. Okay, some cells rarely divide, or they don't divide at all. Yes, Orion, go ahead. You have a question. Sorry, I can't hear you. It's is your mic on? Nope, still not coming through. Maybe you can type your question instead, if that's okay. Okay, great. So while we wait for the question, does anybody have an example of cells that do not divide in the body? Like if we lose it for good. They're not coming back. Anyone have any example of something like that in the body? No, you must have heard about it. Like people say, these cells are very precious in the body, so you better not lose them. I'll give you one example, okay? Heart cells, heart cells. Okay, people who suffer from heart attack, it's hard to recover, right? Because you don't get new cells, you cannot repair the damage, right? And you never heard of people say, you never heard of something called like heart cancer, right? There's no such thing as heart cancer, because if a cell don't divide, it's not possible to form a tumor and therefore no cancer, right? So heart cells, they don't divide. Many people say brain cells and neurons. Yep. So the actual neurons that's responsible for the processing information, the reflex and all that stuff, those neurons, they don't divide. Okay, but you hear people say have brain cancer, right? Like how does that work? Well, there are other types of cells in the brain, in the nervous system, that do divide. Okay, and those are the ones that give rise to cancer. So my original point is that many cells will just be doing its normal function and preparing for the ultimate division. But some cells, they are forever gonna be remaining in the interphase and will never divide in the end. So there's the question about Down syndrome and autism and how the chromosome results in this defect. Yeah, we're gonna get to that. I think later on today. Or actually maybe later. But the short answer to that is for things like Down syndrome, they have an extra chromosome 21, right? And it's not just an extra chromosome 21 that could cause problem. If you have any extra of any of the 22 chromosomes, in fact, sex chromosome can have extra ones as well. That usually most of the time will result in some kind of problems. And missing chromosome can also happen but usually those are more deadly, right? The person will not be able to grow at all. Like it would die in the embryo process. It's the extra chromosome that usually will make it to full term, and that creates a whole set of conditions that we will look at in lecture 8, I believe. Yeah, so hopefully that answers your questions. Good question. Okay, so ultimately you will get to the actual division, the mitosis, where the parent cell, the original cell, would become two genetically identical daughter cells. Okay, so here we go. That's the cell cycle, and this whole thing here is the interphase. That's the normal stage, and most cells would spend like 90% of its time in the interphase. And then this is the actual division, the mitosis, and you can see there are many smaller phases here. Now, within the cell cycle, we have these checkpoints. Okay, checkpoints. Here it says restriction checkpoints, and these checkpoints are basically to determine if the cell is of good quality to go through the division process. So to give you an example, right? Earlier we talked about the telomeres, right? How if it's critically short, then the cell will not be allowed to divide, right? So that would be something that's being checked at one of these checkpoints, okay? If the right here, right, between G1, which is growing, and the S phase, where the DNA is replicated, so there's a checkpoint right here that will see if the telomere lengths are long enough. Okay, if it's not too long, then it will not be allowed to proceed and it will enter senescence. Sometimes it will, if it checks the DNA and there are too many DNA damages, then it will get sent to perform apoptosis. It will trigger cell death in the cell, right? Because there's too much damage. No point of copying this damaged cell. No point of keeping it around. So we might as well destroy it for the well-being of the organism. So there are multiple things that we check for before we proceed with the DNA replication. Okay, and after you copy your DNA, after you do the replication, there's another checkpoint, right, to check for the quality of the clone DNA, right? Did you make any mistakes? Did you copy everything, right? And if all goes well, then you are allowed to go to the final stages of preparation and then finally to the actual division. Okay, so along the interphase, right? Again, you're just growing, doing your own job. Check to see if the DNA is good enough. If it's good enough, then you're allowed to copy it. We check it one more time after you copy it, make some final preparation. Then we go ahead with the division. All right, so 90% of the time is spent in the interphase, and then roughly 10% of the time is spent in mitosis. Now, yeah, these are really rough numbers, right? Don't let them stick in your head for too much. Don't read into it too much. In other words, some cells divide very rapidly, like your skin cell. You replace all your skins in matters of like weeks, right? But some cells they divide very slowly. Okay, and some cells have more regenerative capability than others. Does anybody know which organ in the body can regenerate itself? If you cut off 1/3 of it and donate it to someone, you will get the 1/3 back eventually. It will grow back. Does anybody know what organ is that? Yeah, Andrew said liver. That's correct. Okay, so if you donate 1/3 of your liver to someone else, your own liver will grow back, which is remarkable. Okay, no other organs can do that. Your kidney, you donate a kidney, kidney is not going to grow back, right? Okay, but liver, it's very special. It has the ability to regenerate. That takes us to 9:30. So why don't we take a quick break? 5 minutes, and we will come back and resume. All right. So see you in a bit. All right, let's keep going. So once you've gone through the whole interphase, once the DNA has been replicated and the quality of the replication is good, then the cell is permitted to enter the actual division, right? The mitosis. Okay, so here is an actual picture of what a cell looks like. I just want to emphasize that sometimes you see all these pretty colored pictures, even the ones we saw in the lab, right? Like last Wednesday, the pink and the purple. That's not the natural color of the specimen, right? We added various things to highlight different features, right? We stain them. So here we use various antibodies actually to highlight different parts of the cell, and all you have to know is a blue represents the nucleus, and all the chromosomes you will see later on will be highlighted as blue, and these green lines, they are part of the cytoskeleton. You might remember cytoskeleton. Even I didn't test you on it, right? On test 1. But part of the cytoskeletons involve these spindle fibers, you might remember, that would help guide the movement of chromosomes. And the red is just like the background of the cell, the cytoplasm, if you would. Okay, so at the in the interphase, which is not part of the division, it's just the normal stage, the 90% of the time, right? The cell spends in this stage performing its regular function. But nearing the end of it, we will duplicate the DNA. Okay, so in this case, the DNA is still in its chromatin form. Okay? It's DNA wrapped around proteins. It's not the most condensed version just yet. You might remember these things, right? The centrioles, which are one of the organelles that we've learned, and the purpose of the centriole is to create the spindle fibers that will guide the chromosomes. Now, just introduce a few other concepts and terminologies that you should know. Here, we group chromosomes together, usually not by colors, but by the size. Okay, that's important to understand. In all the pictures, if you see 2 chromosomes that are the same length, that means they belong to the same pair. Okay, remember, you have 2 chromosomes 1, 2 chromosomes 2, all the way to 2 chromosomes 22, and the 23rd would either be identical in female or different for male, right? So these chromosomes, one of them again is from mom and one of them is from dad, and we have a name for them. Okay, these are called homologous chromosomes. Homologous chromosomes. Okay, we will have a more precise definition later on, but right now just think of homologous chromosomes as a same length and size. Okay, one moment please. Okay, so we will have, let's just say this is chromosome number 4. Okay, number 4 from mom and number 4 from dad. So just see if you're with me. How many homologous chromosomes are there in a female? How many homologous chromosome pairs are there in female? Let's do a poll. Is it 22 or 23 in female? Let's do a quick poll. If you think it's A, if you think it's 22, please choose A. If you think it's 23, please choose B. The question is, how many homologous pair are there in female? Okay. Anyone else? Just gonna wait a few more seconds. All right. Most of you chose B. That's correct. Okay, female has 23 pairs. But in males, we only have 22 pairs of homologous chromosomes, right? Okay, because the sex chromosome is X and Y. X and Y are not homologous. They're not same size, same length, right? Right. So there you go. Now, when we copy the DNA, the entire chromosome is going to be copied, and they are going to be stuck together. Okay. Oh, sorry, just going to backtrack a little bit. They are same size and same length, but they are not identical, right? Not identical. I probably should mention that. Okay. What do I mean by that? What do I mean by they're not identical? So you will have a gene here and you will have a gene here. Okay? But those 2 genes they could be the same, but they don't have to be the same, right? Okay. Think about your parents. Right? Your parents obviously are not identical to each other. So, one might have curly hair, the other might have straight hair. So on the mother chromosome, there might be a curly hair gene, right, which will represent by capital C. On the father side, they might have straight hair, which will represent by a small c. So it's the same gene that codes for the same characteristic, but it's a different version of it. Okay. So as another example, right, your mother might have like blue eyes, which will represent with small b, and your dad might also have blue eyes. So it turns out to be the same in this particular case, but for another gene, right, maybe it's different again, right? So you will have the same set of genes, but the versions of the gene might be different. So homologous chromosomes are not identical at all, okay? Because genetically our parents are not identical, right? Okay. So when you make a copy of each of the chromosome, the identical ones will be stuck together. Okay. So the two mother ones will be connected, and the two father ones, the paternal one and the maternal ones, will be connected. The ones that are connected, we call them sister chromatids. Okay, TID. Right? Don't confuse this with the ones that we had earlier, right? The other one is a chromatin, TIN. That's just DNA wrapped around the proteins. But here, the sister chromatids, they are genetically, we'll just say identical, okay? A clone of each other. Replicated DNA, okay? Identical in every way. In every way. Okay? So you start with one chromosome 1 from mom, and now you have two chromosome 1 from mom. And then same thing for the paternal one, for dad ones. Okay? These ones, they are still homologous. Homologous. Okay. But the ones that are connected together, they are sister chromatids. Are we together here? Because it's about to get even more confusing. I just want to make sure there are no questions regarding anything I've said so far. Okay. So hold on a second. Just want to double check something here. Okay. So the location where the two sister chromatids are stuck together, we call that the centromere. Okay, centromere. So there's another centromere here. Okay. Another centromere there. Now, this next part is a little bit confusing for many people. It was confusing for me as well when I was learning it. All right. So let me ask you an obvious question. No trick here. How many chromosomes do you see on the left side here? How many chromosomes? Just type your answer in the chat box. Okay. How many chromosomes do you see here on the left side? It's not a trick question. I just see a barrage of answers in the chat, but there is nothing. How many chromosomes here? Okay, thank you, Andrew. I'm not sure where the rest of you are, but that's two chromosomes right here. Okay, two chromosomes. Two chromosomes. Okay, so after the DNA replication process, if you take a look at here, right? Take a look at here. How many chromosomes are here now? How many chromosomes? Okay, Kavita, Bhavisha, Andrew. Okay, they're saying 4, right? Okay. See, that's what I thought, right? Like logically, like you see like 1, 2, 3, 4, you would think it's 4, right? But it's not, okay? The people who came up with these things, they've decided that, this is still called one chromosome. As long as they're stuck together, we just count it 1. Okay? Yeah, Alyssa, still 2, that's right. So that's 1 chromosome, and that's also 1 chromosome. Do you understand? So as long as they're stuck together, biologists, they still want to count it as 1. Okay. So before replication, 2 chromosomes. After replication, still 2 chromosomes. However, you would have 4 chromatids now. 4 chromatids. 1, 2, 3, 4. Can you guys actually see my cursor? I think you can, right? Okay, so like 1, 2, 3, 4 chromatids, but still 2 chromosomes. Okay, so keep that in mind as we talk about the mitosis, because there will be some questions on the quiz that deals with this. All right. Hopefully that makes sense. If it doesn't, then you know, send me a question. So we're gonna look at the 4 phases, right? Starting with prophase, metaphase, anaphase, telophase. So PMET, that's the order. PMET, prophase, metaphase, anaphase, telophase. All right. So let's start with the early prophase at the beginning of the division. What's happening? The chromatin will coil up, will condense even further to form visible chromosome. So you can start to see the outline of the chromosome, right? The individual strands of chromosomes. Let me just show you the previous picture as comparison. Over here, it's just like, you know, a blue hue. We cannot make out the outline of anything specific, right? So nothing much there. But over here, we can start to see the individual outlines. And that's because the chromatins are now coiling into the chromosomes. By the time you get to prophase, DNA replication would have been completed. Okay. Sorry for like jumping back and forth, but I need to show you what I'm talking about. Look, prophase is right here, right? It's the first step in mitosis. If the cell is allowed to get all the way to prophase, it means it was allowed to copy its DNA, right? So by the time you get to prophase, DNA replication would have been completed already. Okay. So all the chromosomes will have its identical copy stuck to it, and then we would have these sister chromatids held together at the centromere. Okay, so again, just for the sake of visual representation, you have chromosome 1, and then you have chromosome 2 and chromosome 2. After DNA replication, right, they would all be like this now. Do you understand? So this is 1, 1, 2, 2, and then afterwards you have 1, 1, 1, 1, 2, 2, 2, 2, and then 3, 3, 3, 3, all the way to the sex chromosomes, right? Okay. Next, the nuclear envelope starts to disappear. We need to let the chromosome come out of the nucleus so we can move them around. But the nuclear envelope is in the way, right? So we need to dissolve that away, let the chromosomes come out. The centrioles will start to move to the opposite side of the cell, and at the same time, they will create these spindle fibers. Okay, these are little ropes that will eventually help guide the movements of the chromosomes. Okay, so that's what's happening at the beginning of prophase. And then later on, the nuclear envelope is completely dissolved, and you can see the chromosomes are, you know, pretty much out of the nucleus. Right? There's no more nucleus, essentially. It's all in the cytoplasm now. And we can make out the individual chromosomes very clearly. Right? And also the spindle fibers that were produced by the centrioles, I'm just going to write it out for you, produced by centrioles, they will attach to the centromere of each sister chromatid pair. Okay? So remember, you have your copied DNA stuck together at the centromere. So now the spindle fibers from both sides will attach to that particular point. Okay. So for simplicity, they're just showing 4 chromosomes. But in reality, within our cells, we would have 46 of these things, right? Okay, so that's basically prophase. Right? Just to quickly recap, we dissolve the nuclear envelope, we condense the DNA into chromosomes, and then we have the centromeres going to the opposite side. Sorry, the centrioles going to the opposite side, and the spindle fibers will attach to the centromere. Okay. Next, we have the metaphase. Okay, so M for middle. M for middle. In metaphase, everything lines up in the middle. Right? So remember, the spindle fibers from each side will be attaching to the centromeres, right? And that creates a tug of war that will pull the chromosomes back and forth until everything lines up in the middle of the cell. Okay, so that's all that's happening in the metaphase. And in the real picture here, you can see the green lines are the spindle fibers, and all the chromosomes are lined up right at the center of the cell. Next, we have anaphase. A for apart. Apart. So the spindle fibers from each side will pull so hard that the sister chromatids will be separated. Remember, this copy and this copy, they're exactly identical, right? Okay, and this copy exactly identical. So in the end, the chromosomes that are moving to one side, this whole set will be genetically identical to this whole set. And so this whole set will be pulled towards this end. This whole set will be pulled towards this end. Okay, and that's what anaphase is. The sister chromatids are pulled apart and they will move towards the opposite end of the cell. Now I want you to think about this. Look, in metaphase over here, in metaphase, for this particular diagram, how many chromosomes do we see? Okay, just think about it in your head. Okay? You don't have to type it out. You just, based on what I taught you earlier, how many chromosomes do you see here? Okay. So I hope the number that is in your head is 4, right? Okay. 1, 2, 3, 4. As long as they're stuck together, we count them as 1. So if you have 4 chromosomes in metaphase, how many chromosomes will there be in anaphase? How many chromosomes now? Right? They're no longer stuck together. So the answer is 8. Right? Okay. Very good. Finally, we have telophase and something called cytokinesis. T for tearing apart. Okay. So it's actually quite simple. Telophase is opposite of what happened in prophase. Okay. So everything that happened in prophase, we just do it backwards. So now the chromosomes, since we're done pulling them back and forth, they can uncoil back into chromatins. Right? We want to put them back into the nucleus, so it's time to form the nuclear envelope again. The spindle fibers will disappear. We no longer need to pull the chromosomes around. We don't need the spindle fibers anymore. They could go away. And the cell will split in the center. Okay? In a process called cytokinesis, right? Which basically means you divide the cytoplasm equally and the cell will split in the center. Okay, think about like having a big ball of Play-Doh and then you tie a rope around the center and then you pull on the rope, which will cut the Play-Doh in two, right? Okay, or dough or whatever it is. It'll cut it in 2 halves. That's basically what cytokinesis is. Okay, there's going to be some kind of constriction in the middle, and it will cleave the thing into 2 equal cells, right? Each containing the genetically identical DNA as the original cell. So that's basically what happens. Okay, so prophase, P for, you can say, preparation. Preparation. Okay. And then M for lining up in the middle. Anaphase, you pull them apart. And telophase is just backwards of prophase, and you're going to tear the cell into 2 equal halves. Okay, and that's it. That's the process of mitosis. All right, let's take a look at this. This is onion cells. We tried, me and like a few other teachers, we try to use the microscope that you used on Wednesday to show you these mitosis picture, but we weren't very successful because the microscope doesn't have powerful enough magnification. But these are onion cells, okay? And if you take onion cells, look at them under a microscope, you will be able to find all these cells undergoing different stages. So let's see if you can identify them. Okay. This one here, A. What do you think A is? Is it prophase, metaphase, anaphase, or telophase? Look at this one. Any takers here? Okay. Yeah, we have a few people who say metaphase. Yeah, that would be correct because they all lined up in the middle, right? They all lined up in the middle. So this is metaphase. What about this one? What about this one? It's a little bit hard to tell this one versus this one. Okay, but like try to look closely to see I mean, right over here you can kind of see the line in the middle, right? There's no line in the middle here. So this one, yeah, this one is anaphase. Anaphase. Okay. This one you can see if you look really closely, right? You can see a line starting to form in the middle. That's the cleavage furrow in the picture. That's separating two cells. So this is telophase. Okay. And over here, that's the prophase. Okay. Prophase, everything is still like pretty much in the nucleus. You can still make out the circular shape of the nucleus. Once you cannot see the nucleus anymore, then it has to be one of these stages. Okay, so here's another telophase, right? Sorry, another anaphase, rather, anaphase. Right, another anaphase here. And all these ones that you can't really see the chromosomes, all these are going to be in the interphase. They haven't even entered the mitosis yet. Okay. Let's quickly switch to the study guide here. Okay, we got some pictures here. Let's quickly go through them. Okay, I'm gonna give you like a minute or so. I'm gonna give you a minute actually. You can fill it in, all of it, and we'll take it up. Okay. These one are plant cells. The rectangle ones, the circular ones are, animal cells, right? So go ahead, try it out. All right, let's take them up. Okay, over here, we can still see the nucleus, and then we can't really make out the chromosomes. It's still in the chromatin state, right? So over here, we would say this is the prophase, right? Early prophase. All right, next one. Oh, sorry. This is interphase. My bad. Interphase. Because we can't see the chromosomes yet, right? Interphase. It hasn't entered the actual mitosis yet. Okay, let's do all the plan one first. Okay, so this is interphase. Now we can see the individual chromosomes, right? We can see the nuclear envelope is dissolving. We can see a little bit of the spindle fibers, right? So this one is the prophase. This is easy to tell, right? Hopefully, you can see they're being pulled apart. Anaphase. Lined up in the middle, that's metaphase. And this is separating into two separate nucleus, and then you can see the wall starting to form in the middle. That would be the telophase. Okay. This one, right, you can see the two cells are being pulled apart. That is the telophase. Oh, I totally missed this. Sorry. We're going to get to it in a second. What about this one? This one over here would be prophase. Because you can see the individual chromosomes now, right? If you see the X's on the nucleus, it's prophase. If no X, then it's interphase, right? And you can see the centrioles moving to the opposite side. This is the anaphase. This is the metaphase. Okay, and just last one, what is this? Help me out here, please. Anyone? What is that last one? What does it look like? Zoom in a little bit for you. Yep, that is totally anaphase. Okay. So on the quiz, I will only give you cartoon diagrams. Okay, I will not give you real ones to identify. Okay, so make sure you know the cartoon ones, what they represent. All right. Hopefully we'll have the quiz next Wednesday. I'll have to see how much I get done on this Wednesday's class. If we're not able to finish as much as I like, then we might have to move the quiz. I'll let you know. Okay, but you know, according to the schedule, the next quiz is going to be next Wednesday, right? All right, that is that. Let's move back here, and we will continue. So earlier we were talking about checkpoints, right? And it's important to have these checkpoints because we want to make sure the cell DNA has good quality. We want to make sure the cell is in a healthy state before you go ahead with the division, right? Okay. So within the interphase, there are various substages, just like mitosis has the PMET, right? The interphase also has a various stage, right? We have the G1, the S, and the G2, kind of like, you know, getting your driver's license, right? G1 and then G2, and there's S phase in between. Okay, so in G1, again, you're just growing, you're gathering resources, preparing for DNA synthesis, and then there's going to be a checkpoint right here, right, which checks for the DNA quality. If it's good, you will be allowed to copy the DNA. And afterwards, you're going to check again to see if you did a good job copying. All right. And as I mentioned earlier, if there are some kind of errors in the DNA, the cell will try to repair it first. It's not going to kill it right away. It's going to try to repair the error. And if it cannot be fixed, then the cell will not be allowed to divide. And there are a couple of fates to it, right? It might just stay there and enter senescence, right? If the telomeres are too short, for example. If there are too many errors that cannot be repaired, then it will get sent. It will trigger apoptosis, cause it to die, right? And that's the function of the checkpoints. Okay, think of the checkpoint as like you know, green lights and red lights, right? If it's green light, you can go ahead and proceed through the cell cycle. If it's red light, you have to stop, right? And you're not permitted to go ahead with it. Okay, keep in mind this green light, red light situation, scenario or analogy rather, because I'm going to come back to it in the next few slides. Okay, just very quickly, let's do a few polls here. Okay, go ahead. Oh, this question is not even complete. I'm sorry. Let me fix that. During which phase does the nuclear envelope dissolves? Okay, that's the question. All right, I'm gonna close the poll. Any last minute attempt? Yep. Most of you got it right. It's the prophase. Okay, that the nuclear envelope dissolves and the chromosomes are allowed to come out. Okay, here's another one. This is an important one because it's confusing. Okay, so that's what happens in the metaphase, where you have 8 of them all lined up, and then afterwards, they will all be pulled apart, right? All be pulled apart. And then you would actually have 16, right? Okay, so for those of you who answered, most of you got it right. Very good. Okay, this is something that you should think about. We don't have time to go through it right now. Think about it. Maybe we'll discuss it again next time. Okay, so now we're gonna go back to the cell cycle situation. Let's see if I can finish this last part before the end of class. So the checkpoints that occurs, right before and right after DNA replication, right? These checkpoints are actually carried out by proteins. So there are proteins that would check for DNA damage. There are proteins that would check for the telomere length. There are proteins that would try to repair the DNA damage. There are proteins that would trigger apoptosis. Everything is like, controlled by proteins, okay? And you should know by now that proteins are encoded by DNA, okay? So if there is a mutation in the DNA, it's possible for some of these checkpoint proteins to have a mistake, to have a mutation and not function properly. Do you follow my logic here? All these checkpoint proteins are themselves coded by DNA. So if the DNA that codes for them, if the gene that codes for these proteins contains a mutation, it's possible that these checkpoint proteins will not be functioning properly. Okay, that's the premise here. Now, to keep things simple, right? Red light and green light, right? We have 2 types of checkpoint proteins. We have those that are responsible for turning on the green light. We call them growth factors. Okay, so these are the green light proteins. Green light proteins. They are the ones that tells the cell, you are good to go. You are allowed to proceed to cell division. You give them the go ahead, and these growth factors, they promote mitosis. Okay. On the other hand, we have growth inhibitors. Okay? These proteins, they are the ones that are like the red lights. Okay? They inhibit mitosis. You're not allowed to divide because too many errors. Okay? Too many mistakes. Telomeres are too short. Okay? So 2 types of proteins, and they balances each other out, right? Okay, you either let the cell go ahead and divide, or you stop them. And again, all these proteins, they are encoded by DNA, which means they could potentially mutate and not function properly. there are two types of genes that we're going to be looking at. We have what's called proto-oncogene, and the opposite of that is tumor suppressor genes. Okay, a proto-oncogene is basically a gene that codes for a green light protein. Okay, it's a gene is a segment of DNA, right? So that gene encodes for a green light protein. It encodes for a protein that is a growth factor. So normally proto-oncogenes would be responsible for stimulating mitosis, allows the cells to divide and divide. Okay, it's perfectly normal to have these proto-oncogenes. In fact, you need them. If we don't have them, then nothing divides, and we won't be able to grow and develop. So proto-oncogenes codes for proteins that allows mitosis to occur. However, the second point here, when a proto-oncogene mutates just means that there is some kind of DNA damage, right? DNA error. Okay, if a proto-oncogene mutates, or sometimes you have too many copies of it. Okay, remember earlier I was telling you that sometimes it's possible to have extra chromosomes. Okay, like some people might have an extra chromosome 21, right? Well, it turns out in some cases, for reasons we're not going to go into, it's possible to have extra copies of a genes because the segment of the chromosome it's cloned. Okay, you have extra copies of it. So if you have a proto-oncogene that's mutated, or if you have too many copies of it, then this proto-oncogene, which is normally a normal thing to have, is now going to turn bad. Okay, and this bad gene is going to become permanently turned on or permanently activated, which means the green light is always on. The cell will keep on dividing and dividing and dividing, even though it's not supposed to. Okay, because the green light is always on, right? There's no more checks. Even if you have errors, even if telomeres are too short, it totally ignores all that because the green light is always on and it just keeps on dividing and dividing. So cell grows out of control and it could lead to cancer. So this new gene that's mutated, the bad gene, is called the oncogene. All right. Proto-oncogene is the original one. The oncogene is the mutated one, the bad one. Okay. So this new gene is going to code for a wrong protein that's stuck on in the on configuration. Okay. And it can't stop this. Okay? Because it's permanently on, right? Permanently stuck in the green light configuration. Does that answer your question, Orion? Okay, so the opposite is a tumor suppressor gene. Okay, tumor suppressor gene, right? Tumor suppressor gene are genes that normally codes for the red light proteins. Okay, normally codes for red light proteins. When I say green light proteins, red light proteins, like the proper name is growth factor and growth inhibitors, right? Okay, it's just easier to visually visualize if you think about green light and red lights, okay? On the test, please use the proper terminology, okay? Growth factors for green and growth inhibitor for red. Okay, so over here, the tumor suppressor codes for the red light proteins. And normally their job is to slow down cell division, right? Okay, they are trying, these proteins will help try to repair the DNA mistakes, and then it will tell the cell to die if it's irreparable, right? If you cannot repair the DNA damage. Okay, so if the tumor suppressor gene is not working, that too can cause the cell to grow out of control. Okay, this is like having a red light that never turns on. Okay, so the cell is able to divide and divide and divide. Now, there is a very key, but subtle but key difference here of what I'm saying. Okay, the proto-oncogene, when it becomes bad, when it becomes the oncogene, it's a green light that is always on. A green light that's always on. Over here, we have a red light That's not turning on. Do you see the difference? Okay. This is always on, which means it is overly active, right? Overly active. It's always on. This is a red light that never turns on. So it essentially has lost its function. Okay, it's not able to turn on at all. This one hasn't lost its function. In fact, the function is always on, right? So we call this a gain of function mutation. Gain of function. I think it's also coming up in a bit, but let's just write down here. Gain of function mutation. Always on. On the other hand, over here, the tumor suppressor gene, when it mutates, we have a loss of function. Loss of function mutation. Does not turn on. Okay. So remember, we have two sets of chromosomes, one set from mom and one set from dad, right? And here is the normal gene, and you will have two copies of a proto-oncogene. In the same way, you have two copies of the tumor suppressor gene. Okay? And here's the thing. Okay? With the gain of function mutation for the proto-oncogene, only one copy needs to be mutated for it to cause cancer. Okay, so you have two copies, but if only one of them turns on permanently, then the green light is going to be on forever, right? So it's like a stuck accelerator. It will just keep on going with one bad copy. It doesn't matter that this one can turn off. If this one is mutated and it's permanently on, then the green light is permanently on, and it keeps on dividing and dividing and dividing, right? So only one copy of the gene needs to be mutated for the cancer to occur. Contrast this with the tumor suppressor gene. Both copies of the gene needs to be mutated to cause cancer. Okay, so let me just try to draw this out again. You have a tumor suppressor gene here. You have another tumor suppressor gene here. Okay, so let's say this one doesn't work. Okay, loss of function, it mutated. So this red light will not come on, but that's okay. You still have another red light that can turn on, right? Okay, so as long as one of the red light is on, you can still stop the cell from dividing. Okay? It's only when both of them are mutated, then you have no more red lights left. Okay, it's different from the previous situation over here. Right? You have two green lights. Okay, one of them functions properly, it turns on and turns off. Okay, but if one of copies mutated and it's always on, it doesn't matter whether the other one turns on and off regularly. If the other one is always on, then that's what the cells will see, right? And it's gonna keep on dividing and dividing and dividing. Does that make sense? Okay, so I just want to leave you with one final thought before we wrap up. Which kind of genes give you a higher chance of developing cancer? A mutation in a proto-oncogene or a mutation in a tumor suppressor gene? Just one more time. My question is, which one gives you a higher chance of getting cancer? Mutation in proto-oncogene or mutation in tumor suppressor gene? Let's just do a final poll here. Which one gives you a higher chance of getting cancer? Is it A, mutation in proto-oncogene, or B, a mutation in tumor suppressor gene? Okay, I'm just going to wait for a few more responses before I end the poll. Hey, and most of you answered correctly. That's right. A gain-of-function mutation in the proto-oncogene, which turns it into an oncogene. The oncogene is the bad one, right? The proto-oncogene is the normal one, right? So only a single hit is enough to push the cell towards indefinite dividing, right? Going through the cell cycle very, very quickly. But over here, we kind of have an insurance policy. If you damage one of the cell, the gene, if you have a mutation in one of them, that's okay. You still have another one that's going to be working, right? It's only if you have a double hit in the same cell, then we're going to have a problem. Okay, so it's not as easy to turn a cell cancerous if you have a mutation in the suppressor gene, because you need 2 hits in the same cell. Okay, over here, just 1 hit in any cell that could push it over. All right, so keep thinking about that. Make sure you understand the implication of that. And then next class, we're gonna go into more details about how it actually relates to cancer and all that stuff. Okay. So thank you for coming today. I will see you all again virtually on Wednesday. And just to be clear, I got a few emails regarding the lab. You still have to hand in a physical copy of the lab, but that's when we return. Okay. We're going to be returning next Monday to in-class learning. So you can bring it next Monday. All right. Okay. Thank you. See you.